Welcome to Medmarc's webinar series. Today's presentation is titled Medical Device Design and Infection Prevention. What do manufacturers need to know? I'm Kate Klaus, Senior Attorney in Medmarc's Risk Management Department. On behalf of Medmarc and today's presenter, Dr. Howard Loree, thank you for joining us. Dr. Howard Loree has over 25 years of diverse research and development experience in the medical device industry and a hands-on aptitude across the complete product development lifecycle in both startup and growth stage companies. He has developed highly innovative surgical and interventional products for mechanical circulatory support, vascular access, peripheral vascular, neurovascular, spine, and ophthalmology applications. Dr. Loree's consulting activities have focused on cardiovascular devices and include technology assessment, product design optimization, design verification and validation testing, medical device health risk assessment, post-market regulatory support, and assistance with post-market regulatory and compliance inquiries. Dr. Loree has direct experience in leading projects from initial concept through in vivo feasibility, intellectual property development, design requirements, human factors analysis, failure mode and effects analysis, design freeze, technology transfer for product development contractors, design verification and validation testing, and preparation for first-in-human trials. His personal technical accomplishments include raising over $4.5 million in NIH funding for medical device development, planning, executing, and reporting preclinical studies to rapidly establish in vitro and in vivo feasibility, and supporting successful FDA submissions, including IND, IDE, HDE, 510K, and PMA. Dr. Loree's education includes a BS and MS degrees in mechanical engineering from MIT, as well as a PhD in medical engineering from the Harvard-MIT Division of Health Sciences and Technology. At MIT, his coursework and research focused on design, biomaterials, fluid mechanics, and medical sciences. He also completed a postdoctoral research fellowship in cardiovascular biomechanics at the Brigham and Women's Hospital Harvard Medical School. And with that, I am pleased to turn things over to Howard.
Thank you, Kate. It's great to be here. Thank you all for attending. So I hope to, over the next 45 minutes, share some information about how medical device design and infection prevention can work together to create better products and safer products for our patients. And here's an overview of what I'll be covering. First, a very quick background just about me and about Exponent. Then we'll be talking about medical device infections. And I want to promise you, since I know many of you just had lunch, we will not look at any gory pictures of actual medical device infections. We're going to talk principally about the costs and causes, and we'll take a case study approach. And then we're going to delve into some design strategies that we can use for infection prevention, both for single-use devices and reusable devices. And then we're going to close out looking at some of the issues related to device use and the environment, and then how we can do design verification and validation that we actually have prevented infection. So Kate's already mentioned some of my background. These are some of the companies I've been with, and I've recently joined Exponent and really glad to be able to work with clients and helping them to address risk. This is a little bit about Exponent. We're a multidisciplinary engineering and scientific consulting firm. We represent more than 90 different disciplines, and we help our clients by solving technical, regulatory, and business issues. And some of these pictures are projects that we've been involved with. Our company's over 50 years old, started by a group of 5 Stanford professors, and now we're over 1,000 employees, and most of our consultants have PhDs. We're in 30 offices around the world, and we do a lot of different work. You can think of us as a university where if a client has a need, we put together a project, we draw from all the different departments, and we can very quickly address both reactive, such as litigation projects, or proactive projects where we help them to refine and improve designs. So with that, let's talk about medical device infections. The topic of cost, of course, is very important. And when we think about that from a societal point of view, uh, we can begin to understand just how important healthcare associated infections are. Right now in the US, the annual direct medical cost of these infections to hospitals ranges from 37 to $58 billion. And it's thought that if we could prevent those, we might be able to save 70% of that. So the benefit could be 32 to $41 billion. And then a specific type of healthcare associated infections would, of course, be medical devices. And that's accounting for nearly 2 million of the healthcare associated infections reported by the CDC. And those approximately 50 to 70% um, are attributed to indwelling medical devices. So if we were to do all the math, if we did a better job with prevention, um, the costs are ranging 18 to 29 billion. We might save 16 to 20 billion of that. And then, of course, the cost to patients is in illness and death. And the attributed mortality is highly device dependent, but it can range from less than 5% for devices that are simple, like dental implants or Foley catheters, to over 25% for mechanical heart valves. Let's talk about some of the simplest devices first. We're going to talk about catheters, specifically central venous catheters. You can see the picture on the right is showing a catheter that's placed into the right atrium of the heart or nearby, and that allows us to deliver drugs or measure pressures in that region of the body and get it into the central venous system quickly. Right now, there's over 15 million patient days of exposure to these CVCs in the ICU environment. And there's an incidence specifically for non-dialysis catheters of 2.5 to 4 per 1,000 catheter days, where you get a bloodstream infection. That's a septic infection. A specific kind of central venous catheter, the hemodialysis catheter or HD catheter, has an even higher infection rate. And that's shown here on the right side, where we're going directly into the internal jugular vein and then into the right atrium. These infection rates range from 3.8 to 5.5 per 1,000 catheter days, and they become very, very expensive. In the ICU, the cost can be $3,000 per infection. And if they get worse, you're talking about extending the duration of the patient being on a ventilator, perhaps increasing the ICU stay by a week, and then being in a step down unit for another 2-3 weeks. So this is a huge area of concern. And if you know a hemodialysis patient using a catheter, they're often fighting the problem of infection, and it's a very difficult one. This problem of infection is also an issue with cardiac implantable electronic devices. They're called CIEDs. And these, of course, include pacemakers and the more sophisticated products that manage cardiac rhythm, like ICDs and CRT devices. And for these types of devices, the incidence of infection can average less than 2%, but could be even greater, perhaps over 4% for patients that are at high risk. And these will include patients that maybe are coming back in after complications or are immune suppressed. Continuing on with cardiac device infections, these have not only increased over time, but if you pardon the pun, some studies say that they've even outpaced the increase in device implantation rate. So we're having more and more problems, which is probably traced to things like drug resistance bacteria and these patients perhaps being older or sicker with other comorbidities. And these infections are associated with a lot of morbidity, mortality, and prolonged hospital stays. And one of the worst parts is that they often have to go in and take out the device and the leads and try to clean things up and then subsequently reimplant. You can imagine the cost of that. These can be $125,000 to $250,000 costs for an inpatient admission with an extraction. And the mortality is even higher, 9 to 35% in the first year after implantation. So very severe problems, major challenges in managing these infections. And then, of course, one of the things that's been in the news a lot is the problem of endoscope infection. And currently, there's over 500,000 of these endoscopic retrograde cholangiopancreatographies. That's a difficult word. We'll just call it an ERCP, performed annually in the US. And the little cartoon on the right is just showing how a scope is put down the patient's esophagus, through the stomach, into the start of the small intestine, where you can access this little bile duct. And at that point, the endoscope can deploy from a kind of right angle various tools to go in there and sample things or pull things out, take biopsies. And you can imagine it's a very complicated device, but this is a life-saving treatment, and it's very important to patients who are fighting cancer and other conditions that might obstruct that bile duct. Unfortunately, contaminated endoscopes cause more healthcare-associated infection outbreaks than any other medical device. And it's not simply because there's problems with adequate reprocessing. There's cases where it's documented that there wasn't a lapse in the decontamination procedure. So it was something related to the design or the methods that were prescribed for the cleaning. So we can delve into that a little bit here. Looking at the instrument, there's a specific type of endoscope for this procedure. It's called a duodenoscope, and it's among the most complex medical instruments that have to be disinfected between patients. And ever since the beginning of the use of this device, cross-contamination between patients has been a problem. And some of the major reasons for that are just that sometimes people don't adhere to the disinfection guidelines. There's something called biofilm that can form a deposition across the surface, and it's exacerbated by complex geometries, surface defects. And then, of course, the process of cleaning these, particularly in the hospital with staff that may not be thoroughly trained in all the technology, using automated systems can present risks that the device still has contamination on it. And recently, within the past 10 years, there have been outbreaks. So there was one in particular from 2010 to 2015 where more than 400 patients were infected during ERCP procedures. And we've all heard of these superbugs, these terrible bacteria that resist all types of antibiotics. And in fact, this was involved. The most notable was carbapenem-resistant enterobacteria, or CRE. And it's a terrible infection associated with up to a 50% mortality rate. So predictably, there were lawsuits filed against the companies that make these scopes. There was a jury award of $6.6 million, the first of a series in 2017. And of course, it got the attention of the FDA, which ended up issuing warning letters to all three major manufacturers, really for failure to really have an effective post-market surveillance, which we'll talk about. So let's talk about what are the root causes of medical device infection. And I've listed several here. The first two are organisms that originate from the colonizing microbiota of patients or healthcare workers. So in other words, being in the hospital, you're exposed to a pool of bacteria and other microbes. And then secondly, just this general trend towards antimicrobial resistance, where bugs are, there's more and more bugs that are becoming resistant, they're evolving, it's hard to really come up with effective antibiotics. These are problems that we're not going to talk about in our presentation today. They're in some sense beyond the scope of what medical device manufacturers would be able to do something about. But there are several other causes that I think we can do something about. First is ineffective sterilization or poor sterile technique. So this gets down to how do we design our processes so that they're easy to follow, they're done properly. Inadequate cleaning of reusable instruments, to avoid cross-contamination, poor aseptic technique in wound exit site care, where pathogens can enter the device, the tunnel, or the pocket. And then finally, um, the fact that bacteria work in this environment called a biofilm, which they create, and it helps to uh, basically shield them from the action of antibiotics or the patient's immune system. And so all four of these are things that we can address, and we'll talk about as we go on. So we'll start here just looking at some design strategies for infection prevention and looking at some case studies. So one that is very promising and is widely used is what's called an antimicrobial eluting device. So here we show a picture of a little pouch that is used around a pacemaker. This is a product by Medtronic called the TYRX envelope. It's available in Europe. And the way it's designed is you have a bioresorbable material. It's like a suture that is knitted together. And then you coat that with a polymer that's formulated with antibiotics. And then over time, these antibiotics elute locally into the tissue pocket. And within about 9 weeks, by the time everything is healed up, all of the mesh is gone. And so there was a trial, and of course, they always have to come up with a clever name for a trial. This was called the RAPID trial, which stands for Worldwide Randomized Antibiotic Envelope Infection Prevention Trial. You have to kind of be creative with the letters to get that to spell out. And they enrolled about 7,000 patients. These were high-risk patients. They did a 2-arm trial where they used a control versus this mesh, followed the patients for a year, and you can see there was a significant improvement in infection rate, which dropped from about 1.2% to about 0.7%, and it was highly significant. So that's an example of how a fairly simple technology can make a difference in preventing infection of an implantable. Here's another example of something. This is an antiseptic eluting device. So an antiseptic is not necessarily an antibiotic. It's not a drug. It's really more of like a detergent or a solution that can kill a wide spectrum of organisms. And an example is the BioPatch by Ethicon. This is a division of J&J. And it's just a little disc that's made of urethane. And after you put in a catheter or any sort of a line, you just take the disc and you stick it down. It has a gap on the edge so you can put it right over the catheter. And that stays in place within over 7 days, and it elutes chlorhexidine. And the results of that are pretty impressive. That in the trial that J&J did, they saw 60% reduction in catheter-related bloodstream infections. These are the really severe septic infections versus standard of care, and a 44% reduction in local infections. And this can be used both with vascular and non-vascular devices that cross the skin. You can see many examples. So this is a product that is sold worldwide and is a very popular solution to helping to manage these cases. We can also rely on what are called hydrophilic coatings. And these are just coatings that are very slippery. And one example is the Surmodic Serene coating, which I've actually worked with. It's a covalently bonded UV cured formulation, and it's put on in 2 layers. So you have a base coat shown up here in the upper right that bonds it to the substrate, and then you put the top coat above it. This allows it to stick to various polymers and a wide variety of other substrates. It's a very thin coating. It's very low friction, very low particulates. They're very popular in catheter systems where you have a lot of things sliding and need to be very low friction. But the goal here is that it reduces bacterial adherence, really prevents the formation of that biofilm. And of course, you can formulate it with different antimicrobials or even antithrombotic agents. So there have been studies that have been done showing the effects of these hydrophilic coatings for various urological implants. There can be up to a 50% reduction in infection. And interestingly, you can go online and you can see that a lot of urinary catheters, particularly intermittent catheters that are put in when you need to drain the bladder, are offered with hydrophilic coatings. Although there are no studies that really show that's efficacious, but maybe there will be someday. Another example that is in the realm of tissue engineering is the cuffed and tunneled hemodialysis catheter. And this looks a lot like the other catheters I showed earlier, central venous catheters. What's unique about it is that you have this little white cuff, you can see it here in the lower right, that's made up of a polyester velour. So this is typically, trade name would be Dacron. And the idea of it is that you can reduce the rate of infection by putting this little cuff maybe about an inch below the skin level, and the tissue will grow into that over time. And it acts to seal off the catheter tunnel and prevent bacteria from working their way down from the skin into the venous circulation. Now, of course, you're not going to put in something that promotes tissue ingrowth if it's just temporary. This is something that would be for intermediate or long-term vascular access, on the range of months. But these devices are popular and they do help to control infection. Although I would say that a hemodialysis catheter doesn't have a great track record with regard to infection anyway, but it does offer some improvement. All right, some other more sophisticated examples of design to prevent infection include the wireless pacemaker. And these are just coming onto the market. And you can see here in the lower left a picture of one of the devices. This is the Medtronic micro device. It's 90% smaller than a typical transvenous pacemaker. It's placed directly into the right ventricle. You can see the picture here on the right. And the advantage is that it eliminates various complications associated with a traditional pacemaker with leads, namely pocket infections, hematoma or bleeding, bruising, lead dislodgement, and lead fracture. So these devices currently are limited to right ventricular pacing. So they're not offered, with all the other features like cardiorevascularization. But I expect that they will become more sophisticated, and I expect that we will begin to see more long-term outcome data on them. But it certainly is an attractive design with regard to reducing just the general surface area of biomaterial and different access points where bacteria can enter the system. Another thing that the FDA has weighed in very heavily on is the design of reusable endoscopes. And I'm just showing a picture here on the right, just to give you a sense that the tip of an endoscope is an extremely complex structure. And you can see there's lots of junctions between different materials. There's nozzles, there's ports where air can come in and out. Obviously, there's the optical system. So the FDA looked at all these adverse event reports and information about designs and tried to figure out, well, where are the bacteria hiding on these devices? And predictably, they said, well, it's clearly a risk to have long, narrow channels, especially with sharp edges or rough surfaces. Any sort of hinge, sleeve, any gaps between different parts, O-rings are a great place to trap bacteria, valves, and any structure like this that you can't easily take apart for cleaning is going to be a problem. And so that should be avoided in a design of this type. And, from the earliest stages, the manufacturer should consider how they can replace these features that are challenging to reprocess with something that's single use, and putting in flush ports and just providing accessories that help to clean the devices. Here on the right is probably the biggest problem with a reusable endoscope, and this is what's called the elevator channel. So at some point you have to get this device, which is along the axis of the instrument, to go more or less perpendicular to it, to dive into that biliary duct. And this is an example of a biopsy tool, and this little cable is pulling up this elevator to push the thing out. And all this, of course, is exposed to the environment of the intestine, and all sorts of debris and stuff can get in there. So it really is a very difficult design problem. And all these have to be addressed in a way that we try to minimize sites where contamination can occur. Of course, the solution that would be ideal is to just throw away the endoscope so it's never reused. And in fact, there are several companies that have FDA cleared products for this purpose. One of them is the Ambu, which is sold in the configuration for as a duodenoscope. You can see it's just packaged sterile. You just tear it open, everything comes in a little kit, and it pretty much looks like a standard endoscope at the handle and at the distal tip. So I believe these are going to gain traction. I think that just with regard to design, it's a terrific solution to the problem. Okay, we're going to shift gears here a little bit just to talk about single-use devices. So if we have a single-use device, then obviously it has to be delivered sterile. And there's a couple of ways to think about sterilization. First would be with a gas. And the advantage of gas sterilization is that it's very low temperature, but it requires packaging and a design of a product that allows it to be permeable to the gas. So we've probably all heard about ethylene oxide. This is a very potent sterilization agent. It's able to penetrate multiple layers of packaging. It's great for devices like catheters that have lots of nooks and crannies. It's compatible with most materials, has a fairly short cycle time of days, but as you may have heard, it presents an environmental hazard. So they found, high rates of cancer around these plants that do the ETO sterilization. And so there's a lot more pressure on from the regulatory agencies. And in some cases, it's difficult to find vendors that are still doing this. So there are delays. There's also hydrogen peroxide. This has a more limited penetration, but it's a great solution because the only residuals are water and oxygen. It has a very short cycle time of hours, but you can't have any moisture in the device before it goes into the process. So it's only for dry components. So those are two popular ways of gas sterilization. There's also the use of radiation and heat. And these are very short, very quick processes, but they have limited material compatibility. So if we're to look at gamma radiation or X-rays, you can get a very precise control of the dose and the penetration. Of course, there's no chemical residuals, very short cycle time in hours, but it's incompatible with certain polymers, acetyls, Teflon, any polypropylene that's unstable. And if you don't have the right additives, you will get a discoloration of polymers like PVC or polycarbonate, and that may not be attractive in your product. There's also steam or dry heat. This has a very low processing and capital cost. So of course, this can be done in-house and very short cycle time. You can use it on materials that are made of, such as glass and metal. So of course, pharmaceutical vials are done this way, surgical tools in the hospital, but clearly you can't put electronics into a autoclave or any sort of complex assembly with multiple materials where there's different rates of expansion. So there are, some great ways to sterilize things. Each of them has specific advantages and disadvantages. All right, so we've talked a bit about what's going to happen with the design in getting, dealing with the materials or the design to prevent infection. But device use and the environment is also critically important. And so there was a big question that was coming up back in the late 80s, early 90s, which was, why are we getting cases of adverse events such as infection in hospitals? And there was a study done out of Harvard that looked at 30,000 discharges from over 50 hospitals in New York State in the early 80s. And what they showed was that adverse events, which was defined as prolonged hospitalization or disability at the time of discharge, that of course didn't exist prior, occurred in almost 4% of hospitalizations. And when they looked at these adverse events, they could say that about 58% of them were due to errors, some sort of mishap in the process in the hospital, and 28% were due to negligence. And most of these adverse events gave rise to disabilities lasting less than 6 months, but almost 14% resulted in death, and about 3% caused permanent disabling injuries. And notably, drug complications were the most common type of adverse event, almost 20%, followed closely by infections, in this case, wound infections, 14%, and other technical complications. So what this is just suggesting is that most of the problems, over half of the problems with infection, are probably due to an error in the way the device was used or handled. And so what could we do to address that? And this brings in the role of human factors. So if we're to accept the hypothesis that use errors are often attributed to the design of the device or process itself, then for our devices, for medical devices, we can use tools such as human factors and usability engineering to focus on the interactions between users, the use environment, and the device itself. So the goal, of course, is to minimize use-related hazards and risks, and then somehow measure to confirm that users can use the device safely and effectively. So let's look at a couple examples in this space. So the FDA very quickly began to key in on this with regard to reusable duodenoscopes. And so shortly after this epidemic of cases of infection, they convened a panel and they sought advice about how to better do reprocessing of these instruments. And these were just quotes from their findings. It is important to consider the device, the end user, and the use environment when developing reprocessing instructions. And human factors testing plays an important role in ensuring that end users will be able to understand and correctly follow the reprocessing instructions in the labeling. And based on what this panel recommended, the FDA then began to require human factors as part of any pre-market assessment of a product. So it's very much mandated now. So we can talk briefly now about design verification and validation for infection prevention. Of course, back to the theme of duodenoscopes, the question is, how do we ensure that user materials, training materials included with the instrument, labeling and instructions for use are sufficient to ensure user adherence to the reprocessing instructions? And the answer is, well, we have to put users in a simulated use study and make sure that they can succeed in what the manufacturer intends for disinfection. And shown on the right are just some typical operations that are going on where you get the instrument out of a case, you have to clean it, you have to then prepare it to go into an automated machine that does the cleaning and disinfecting. And these participants in these studies need to be representative of the staff that would actually be performing these procedures. So that's part of what we call usability testing. Then, of course, with sterilization for single-use devices, the FDA has long been in the business of providing guidance on how to sterilize devices. Currently, there's sets of recognized standards for sterilization validation, whether you're using ethylene oxide, radiation, or steam, to just show a few examples. And practically, this sterilization validation is going to occur after the manufacturer has designed the device and the packaging and finalized those designs. And it's often outsourced to specialized contract laboratories, and it's typically done along with a suite of testing related to bioburden and pyrogens. So these are all important things to plan as we develop medical devices. So I can offer a few conclusions from what we've looked at today. First, medical device infections do lead to significant rates of patient morbidity and mortality. They place an enormous cost burden on our healthcare system, and they expose manufacturers to the risk of product liability litigation. And these infections can be minimized by good design practices, including human factors and technologies that inhibit bacterial growth on implanted devices. And finally, design verification and validation of infection prevention is necessary for both single-use devices, in other words, sterilization validation, and reusable devices, essentially usability studies of reprocessing. So thank you for your attention. I'll be happy to take some questions and really appreciate you being a part of our webinar.
Thanks very much, Howard. We do have a few questions for you. First, what is involved in conducting a human factors analysis and usability study of a medical device?
Sure. So I've been involved in several of these, and basically what you would do is try to identify all the tasks involved in using your device. And then what are the risks associated with those tasks? So you're sitting down with a group of people, usually different users and people who designed the instrument, and you're trying to identify every step that people need to do properly to use the product safely. And then once you've figured that out, then you bring in study participants who represent these end users, and you actually observe them using the product in a laboratory setting that simulates the real world. And they actually have, test centers where you can do this, and you have one-way glass, and you can observe them and record them. And if you see an error on a step of use, then you begin to analyze that, and you think of ways that you can reduce that risk. And then you go back and you repeat the analysis. And of course, the FDA has a lot of guidance on all of this for example, reprocessing of devices and just in general, the design of any medical device. So it's a very well-established process. We do a lot of this at Exponent. There's a lot of other providers as well, but it's certainly something that should be a part of every development project for a med device.
Thank you. Our next question, what role could tissue engineering play in improving the infection resistance of an implanted medical device?
Yeah, that's a great question. It's funny because tissue engineering sometimes gets a bad rep that it's the greatest technology of the future, and it never it never actually comes to reality, but actually it is coming to reality. There's a lot of applications of it. One of them is just taking materials that are native to the body, like collagen. And that's a terrific substrate for a scaffold to help tissues grow into. We might also use things like growth factors or cytokines. These help to stimulate cells to do various things and create a construct that then just triggers the body's natural healing process, which includes a mild immune response, an encapsulation of the device, proliferation of the cells, and then ultimately new tissue generation at the interface between the device and the body. And of course, it's this tissue ingrowth which serves to inhibit biofilm growth on the device surface. So there's a lot of applications of tissue engineering, and it's also called regenerative medicine. It's something that holds a lot of promise for preventing infection.
Thank you. What are the FDA's primary concerns when reviewing the infection resistance of a medical device?
So there's three parts to the answer. First, the FDA expects that the manufacturer will address infection resistance at the earliest stages of the design process. And this is all part of risk management. So we identify risks and then consider how to mitigate them. There has to be a biological safety assessment, and then a lot of testing to ensure that any risk related to infection is identified and minimized or mitigated. Secondly, it's critical that the manufacturer has an effective post-market surveillance plan. And post-market surveillance just means that there's a set of activities conducted to collect and evaluate experience gained from medical devices that have been put on the market, and then to identify the need to take any action. It has to be structured and powered to identify problems with infection. The manufacturer has to be looking for the right signals, and they must continuously review their findings, assess what is and isn't relevant, and then respond accordingly. And then thirdly, the manufacturer has to have a quality management system and procedures that are ensuring that everything is well documented. So if the FDA asks questions, then the conversation is about what you did rather than what you didn't do but should have done. So just, in summary, you have to do the right testing and mitigate risks. You have to vigilantly survey the landscape of actual device use, and all the while have a strong quality management system around these activities.
We have just one last question. You mentioned in your presentation regarding scopes that single-use scopes have been introduced to replace scopes that were formerly reprocessed. Is there any concern about a loss in quality, because a single-use device is obviously going to face more pressure on its pricing? And how can companies balance the need to sell their product at a reasonable price, but also provide this safer preventive technology?
Yeah, I think it's a great question. So, we've all heard that term disruptive technology, and it often is referred to as, something new that comes along. And initially, it doesn't quite work as well or doesn't provide all the features of what's already being used. Okay? But in certain niches of the market, let's say for a simpler procedure, it really begins to demonstrate its potential. And then technology continues to evolve, and we can work on ways of making it better, cheaper, to expand into more of the market. So I think that's probably going to be the approach with single-use endoscopes. And I think it's obviously a question of looking at the whole picture of cost. So it's not just what is the cost of the device that you could be using, whether it's reusable or disposable, single use. It's, what is the cost of any reprocessing? What is the cost of repairing the device if it has to be reused? What are the costs of having to treat an infection that might result from it? So, in other words, trying to find ways of justifying a higher cost for a higher quality disposable. But I think it's a great question. I think these devices are just starting to hit to market, and it'll be interesting to see what the adoption is, and whether the users are going to, accept the level of quality that you can get in a disposable, and what they need to do to make it a real winning product. But I think it's inevitable that it has to go that way, in my opinion.
Howard, thank you very much for your time today. This is such an important topic, and I know your presentation will be valuable to our audience. Thank you for joining us. This concludes today's webinar.